Hello, and welcome to the first installment of the podcast series On Air. We are very excited to embark on this journey, and we look forward to exploring the clinical applications of immune cell repertoires. In this series, we would look at how T and B cell receptors are currently used in diagnostics. We will discuss different areas where repertoires can be a great addition, and the reasons why we are just not quite there yet. The podcast series is supported by the Antibody Society. Society aims to bring together everyone working with antibodies, and you can find out more information about the Society on the website antibodysociety.org.
This podcast episode is hosted by Ulrik Stervbo and by me, Nidhi Gupta. I'm very happy to introduce our first ever guest, Nina Luning Prak from University of Pennsylvania. Hello, Nina.
Hello, Nidhi, and hello, Ulrik. It's a great pleasure to be here today.
Nina, you're in Philadelphia. If I have only half a day and I have already seen Liberty Bell, where should I go next?
Oh, there's so many possibilities. Well, it really depends what you like to do. If you're a frustrated sports fan, then maybe there's a game that's happening with the Philadelphia Phillies, which is our baseball team, or the Flyers, our hockey team, or the Eagles, our football team. They are all in stages of rebuilding at the moment, and it's very frustrating for people who do like these teams. But maybe they will be back to their winning ways one of these days or decades. Or you could go to the art museum, which is quite nice. Or you could enjoy maybe a concert. Philadelphia has a very nice music school, and it also has a beautiful concert hall and a pretty good symphony orchestra, actually. So maybe those are some possible things you could see and do in Philly.
So quite a lot, apart from the always present Liberty Bell, I guess.
The Liberty Bell and Independence Hall are certainly staples of Philadelphia.
Oh,
and the university where I work is called Ben Franklin's University. So it was founded by Ben Franklin, and there's a big sculpture of him in the center of the main square of the university.
Oh, definitely worth a visit, I am sure. And speaking of the university, so before we actually get into your fascination with B cells and the B cell receptor, we should maybe talk a little bit about the AIRR-Community, because it's essentially what has brought us all three together today. And, well, not just because of that, but also because this podcast is the work of the diagnostics working group of the AIRR-Community, which is a relatively new addition to the diverse areas covered by the community. So maybe you could tell us a little bit about why was the community formed in the first place?
Sure. Well, maybe if you don't mind me taking a step back and starting with what actually is the AIRR-Community. So the AIRR-Community is, AIR stands for Adaptive Immune Receptor Repertoire, and that refers to antibodies and T cell receptor gene rearrangements. And so the AIRR-Community technically is actually a committee within the Antibody Society whose mission it is to advance and promote the generation, analysis, standardization, and sharing of high-throughput sequencing data that characterize adaptive immune receptor repertoires, also known as AIR-seq data. It's really a grassroots organization that has a variety of different stakeholders, including wet and dry bench scientists, computational biologists, software engineers, ethicists, and even legal experts. And the AIR-Community is open to anyone who's interested in immune repertoire profiling. As far as the origins, it really dates back to 2015 when there was or maybe even earlier than that, actually. But in 2015, there was a meeting of individuals who were interested in immune repertoire profiling in Vancouver. And the organizers of that meeting were Felix Braden, Jamie Scott, and Tom Kepler. And they really spearheaded the initial kind of organization of the AIR-Community and got a whole bunch of us excited about joining this group and working together on improving things like data standardization, optimization of experiments, software, and so on. And so after 2015, the community continued to grow, and we started to form these working groups, which are really, and you've already mentioned, Ulrik, the diagnostics working group, but these are kind of freely formed entities that the AIRR-Community periodically reviews in its annual meetings. And currently we have several working groups. So we have the MyAIR Standards Working Group. We have the Biological Resources Working Group. We have a Common Repository Working Group, Data Representation Working Group, the Diagnostics Working Group that you mentioned. There's also the Germline Database Working Group, Legal and Ethics Working Group, and Software. In addition to working groups, the AIRR-Community also has 3 subcommittees, and these are groups that, so the difference between a working group and a subcommittee is that the working groups form to try to complete a specific task, and that task is kind of reviewed and vetted during the annual meetings of the AIRR-Community. The subcommittees are more involved with ongoing work that will extend beyond a single meeting cycle. So those are the communications subcommittee, the meetings subcommittee, and the inferred allele subcommittee. And then overseeing both the working groups and the subcommittees is the AIR Executive Subcommittee. And then the AIR Executive Subcommittee in turn, and the AIR-Community itself actually in turn, is a committee within the Antibody Society. So that's a very kind of long-winded answer of what is the AIR-Community, how did we form, and what are the various components of the AIR-Community.
So you have an interesting background because you studied molecular biology, but then you went on to obtain a PhD in immunology together with an MD. Where along this line did you become attracted to B-cell receptors?
Oh, let's see. The obsession with B cells started at an early age. I guess I would have to say probably during my PhD training. I worked in the laboratory of Martin Weigert, who is a B cell guru, of course. He contributed many things to the B cell literature, for example. He was was the first person to describe somatic hypermutation of immunoglobulin genes. He also, incidentally, before that, defined stop codons in the genetic code. He has way more Nature papers to his name than I will ever have. And he also showed that somatic hypermutation contributed to selection of clones. And when I was in his lab, we were working on the phenomenon of receptor editing. And so this is where a B cell that has an autoreactive receptor can change the specificity of that receptor by undergoing further DNA rearrangement and convert from an autoreactive receptor to a potentially non-autoreactive one, and thereby survive negative selection. So yeah, I think that was where things started. And when we were studying receptor editing, it was really not receptor editing that we were intending to discover at all. Rather, we were trying to figure out how to model autoimmunity in mice. So the way we did that was to create a transgenic mouse that had an anti-DNA antibody. And we thought, hey, well, maybe this mouse will get lupus, right? This was the naive idea. It was like, well, so, people with lupus have anti-DNA antibodies. Anti-double-stranded DNA antibodies are pathologic hallmarks of systemic autoimmune diseases such as lupus. And so we thought maybe the mouse would come down with, nephritis or other features of lupus. But this never happened. And the reason it never happened was that the antibodies found a way to escape through editing. So these were not your conventional transgenes. They were actually knock-ins through homologous recombination. So the locus was available to RAG to undergo further gene rearrangement because it was knocked into the immunoglobulin locus. And so we defined a variety of different gene rearrangement pathways, primarily in the light chains of antibodies that would modify their specificity. And that was how the mice managed to escape from having these nasty anti-DNA antibodies. They would change them. And from there, I took a brief break from B-cell immunology, did some clinical training, and then wound up training in pathology, where I could again return to my love of B cells, since there are plenty of diseases that have B cell contributions, both in autoimmune disease as well as in malignancies and other difficulties. So that was a way to then try to combine some of my interests in the more esoteric features of antibody gene rearrangements and the more mainstream features of diagnostics.
Mm. And what is it about B cells that still fascinates you?
Oh, I don't know whether there's just one thing. So, I mean, B cells are really interesting because they are quite diverse in their types. So, of course, as B cells develop first in the bone marrow and then later in the periphery, in different tissues in the body, they adopt many different forms. And so, in the bone marrow, of course, you have developing B cells. You have to undergo this incredibly intricate program of gene rearrangements to generate the antigen receptor. You have to select for B cells that are, that are not autoimmune or in some other way pathologic. And then in the periphery, you have to expand selected clones of B cells to participate in immune responses. And eventually, you even have to create B cells that mature into terminally differentiated antibody secreting cells or plasma cells, and all these different subsets of cells are complicated. They work not by themselves, but in combination with other cells of the immune system. The complexity of it is really quite fascinating, and the complexity of looking at an immune response is fascinating because it's not just one cell; it's a collection of cells or clones that get recruited into a productive response to infection, and that whole process can be can go horribly awry in diseases such as autoimmune disease or cancer, and so I think that's another feature to B cells that make them so fascinating to study. There are many different angles that you can look at, both from protective immune responses to maladaptive immune responses. And also just their developmental trajectories, their tissue-based responses, which is really most of what our immune system is doing, right, is responding in the tissues. But we rarely look in the tissues because we often don't have access to the tissues. At least in clinical work, we're often kind of limited to the blood or to easily accessible places. So, yeah, so I think I'm sorry it's not a simple answer because there's so many opportunities for exciting and interesting things.
It is fascinating.
So you mentioned that pathology allows you to, or makes it possible for you to mix B cells with your clinical training. And you are now indeed professor of pathology and laboratory medicine. And it's not a coincidence then that you're our very first guest here on the podcast, because who else would be better to help us kick off this podcast where we try to look at repertoires and clinical application and diagnostics. And I know among you do work on many different things, but some of the things you work on, I know, is the diagnosis of T and B cell malignancies and also on minimal residual disease evaluation. Can you take us a bit through the process in the laboratory? So what happens from a sample arriving until you hand out a result, and how do you evaluate the results at the end? That's perhaps more interesting in this case. So what happens with the sample?
Yeah, I mean, it's a great question. So let me just see if I can clarify a little bit. So are you speaking specifically about a clinical sample where you're evaluating, let's say, a patient for the presence of a malignancy?
Yeah, that was my idea. So
okay.
A clinical sample for this. Exactly.
Right. So, I mean, I guess the first point there is it's not a sample that's in isolation. So when you're evaluating a patient for a malignant condition, you're usually looking at a whole series of different test results, anywhere from, a a biopsy, a tissue biopsy, let's say if somebody's being evaluated for lymphoma, let's say, maybe radiology, CAT scan, PET CT scan, something like that. And then, of course, a lot of clinical lab tests as well, for both for diagnostic purposes, but also for staging purposes and figuring out what kind of therapy someone will receive. So none of these tests is sort of done in isolation. But I think steering the conversation a little bit more towards the AIRR-C side, why don't I walk you through what would we do with a blood sample, let's say, where we're evaluating somebody for the presence of a clonal expansion in the blood, but also kind of, again, take a step back that in quote unquote real life, that there are multiple tests that are being run, and so the AIRR-C tests would be a complement to existing tests that are also being performed, primarily actually on the biopsy sample initially to characterize the malignant clone, and you can use the biopsy of the potential malignancy to define the clonal expansion. So the clonal expansion, what do I mean by that? So if you take a tissue sample and you sequence antibody gene rearrangements from that tissue sample, you will obtain a variety of DNA sequences that correspond to antibodies. Typically, people use the heavy chain of the antibody. And the reason they do that is the heavy chain is the most diverse part of the antibody. So usually you take primers that are flanking the CDR3 sequence. So antibodies are made up of complementarity determining regions, or CDRs, and framework regions, or FR or FWR. The primers normally that we would use in a clinical setting would be in framework 1, which is sitting at the sort of 5' end of the variable region gene, and a primer in the J region. So those primers would flank most of the antibody V gene, including this third complementarity determining region, or CDR3. So CDR3 is made up of the V variable diversity and joining gene segments that come together during VDJ recombination. And that is an extremely diverse part of the antibody because you have different V genes, different D genes, different J genes. So that's combinatorial diversity. You also have junctional diversity. So even for the same V, D, and J gene, you don't get the exact same sequence in two independent rearrangements because the junctions between the recombining gene segments can vary. So you can have additions of nucleotides called N additions, or you can have deletions, exonucleolytic nibbling. You can also have additions through the recombinase, the RAG enzymes that put the rearrangements together. So you have junctional diversity as well as combinatorial diversity that contribute to this extremely diverse CDR3 sequence. So why am I rambling on about this so much? Well, so the CDR3 is very useful as kind of a fingerprint of a clone. So basically, when you sequence all these gene rearrangements for antibody heavy chains, you get a bunch of these CDR3 sequences, and then you make a statistical argument. You basically say, if you have a certain V, D, and J gene, and if your CDR3 sequence is, let's say, 85% similar at the amino acid level to another VDJ that's the same 3 VDJ, maybe that's part of the same clone, and it's also the same length and things like that. And why would you say that? Well, because it's very unlikely to get sequences that are so similar to one another and yet so diverse to arise independently by chance. So you use this CDR3 sequence as a clonal tag, and then what you do is you sequence all these antibody heavy chain gene rearrangements. You take all the CDR3 sequences and you align them into the highly related sequences or clones, or sometimes these are called clonotypes because you're doing it kind of computationally. Whereas a clone is sort of the Greek ideal of the ground truth. Like the clone technically, of course, is clonally related cells are those that came from a common progenitor. But again, in these sequencing data, we're arranging these related sequences together into clonotypes. And then we're making some claims about the distributions of these clonotypes in the underlying population or group of cells that we've sampled. So getting back to the tumor, if we take a whole bunch of cells from a tumor and the tumor has, let's say, one huge clone in it, then what you will see is an extremely dominant representation of that single CDR3 string. And then maybe you'll see a couple other sequences, but let's say 99% of the sequence copies in your sequencing library are going to be that one clone. So again, you amplify the rearrangements, you put them in the sequencer after you add some adapters, and most people do this on an Illumina platform. And again, in the clinical lab setting, most of us use genomic DNA because it is very easy to use. It's a single template per cell, and it's parsimonious in terms of workflow, much easier to work with than RNA, and it's also less subject to differences in transcript abundance and things like that. So usually we use DNA. So then you get all these sequences back, and then you look for these large clonal expansions. And if you find them, and we can argue about how to define them and all that, but if you find these large clonal expansions, then you have a very useful piece of information, because in addition to now knowing what the patient's clone is, you can track it. And the reason you can track it is that you can then take other samples from the patient, either, say, for instance, the blood from a patient with lymphoma to see if there are lymphoma cells circulating, or you can take blood from a patient after they've received treatment and see whether the clone has now disappeared or whether it has come back. And this type of monitoring is something that we do for minimal residual disease, and that's something that you can use to guide therapy. I think that sort of takes us through most of your question. Sorry for the long-winded answer.
It's also a quite open question, I guess, as I realized most of my questions are. And also this next one that I have prepared for you. So, because you recently had a paper out in the journal eLife on biological standards for AIRR-C. And can you maybe frame this paper for us in the context of diagnostics or any clinical setting?
So the paper in eLife was, I do want to give credit, especially to the Biological Resources Working Group of the AIRR-Community, who were the primary movers and shakers to create that paper. And I think, there were, and I'm actually just pulling it up on my desktop here just to look at it real quick. So just so that people know, it's the paper, first author is Johannes Ulrik. And the paper is called Biological Controls for Standardization and Interpretation of Adaptive Immune Receptor Repertoire Profiling. So the aim of the paper was to try to sketch out, what kind of controls currently exist for performing AIRR-C experiments, but also trying to define what are the needs for, so we have certain controls that exist, but there's a lot of controls that don't currently exist. And so what would be the ideal controls that one would like to have for AIRR-C experiments? And so, just in a general sense, controls have a lot of different uses. So you some of the things that you might be interested in controlling in an AIRR-C experiment are how sensitive is the assay? So, and so a kind of control that you might use for that is to do a spike in or a titration experiment where you take a known sequence and you dilute it serially into a polyclonal sequence of rearrangements or polyclonal series of rearrangements. But you might also want to know things about amplification bias. Are the rearrangement frequencies that you are looking at in a repertoire really what you see in the underlying population, or are they a consequence of the way in which you've retrieved the sequences somehow, either in how you've amplified them, or in other ways how you've recovered the different cell types? Maybe one cell type is more easily recovered than another one. So there are these sorts of reasons for disconnect between reality and what you obtain. There's fidelity in sequencing, which is another potential issue. So different methods have different error rates associated with them. So, for example, with a DNA-based method, you have kind of your initial template, and that's it. You sort of have one shot in a way. Whereas if you do things from RNA, you have multiple shots on goal in a single cell, for instance, because you have several templates, and you can sequence, you can generate cDNA from the RNA, and then you can even do this handy trick where you do molecular barcoding, where at the cDNA synthesis step you would include, an adapter or a short sequence of variable nucleotides at the cDNA synthesis step. And then every single PCR product that you amplify from the cDNA that has that same molecular barcode, you can use that to then create a consensus sequence of sequences that share that barcode. And that would be a very high fidelity sequencing method. Even though the sequencing method itself is not super high fidelity, by doing this little computational trick, you can create a much higher fidelity sequence. And that might be useful for certain applications. There's also just things like yield of sequencing, copy number skewing. I mean, there's a lot of different things where you might want to have better controls. And so in this paper, we kind of sketch out different types of methods for sequencing. So we talk about bulk gDNA sequencing, we talk about bulk cDNA sequencing, and then also single cell cDNA sequencing. And then we review some of the different controls. And we also survey the community for their interests and their use of different control types. And it's actually quite interesting. There's a broad range of use cases for some of these controls. And I think we got quite a variety of responses in our survey. And, in the end, you look at the different uses. It's, you got sequencing errors, sensitivity, specificity. Another one actually that I didn't mention was contamination. So detection of contamination, which is a huge issue potentially, right, for sequencing, and certainly something that in the clinical lab you want to avoid at all costs. Especially if you're doing MRD analysis. And so there, we worry about that and maybe try to use different barcoding primer sets and also put the diagnostic sample on different sequencing runs from the MRD sample and things like that. You have issues of sample quality control. You have batch effects, which also are potentially significant, linearity and accuracy of clonotypes, and then a whole series of data processing things to consider as well. So I could go on and on about any one of these, but I think for clinical applications, probably the most important are sensitivity, contamination, and to some extent batch effects. Specificity is less of an issue if you're looking, if the sequence that you're trying to find back in a patient is sufficiently unique. And so by that, I mean, if you're doing clone tracking, for example, what you'd like to have is a sequence that has a reasonably long CDR3 string, and ideally is not like a super common V gene, so that the chances of finding it back in an unrelated cell, but then calling it part of the malignant clone, so that the chance of making that error is hopefully not very high. If somebody's malignant clone has a very vanilla sequence, then this method will not work well to track it.
So about the sensitivity, is this necessary to have in each run? So a control for this, or isn't this rather something that you would assess and define during the setup of the entire process?
Yeah, I mean, it's a bit of a loaded question, because on the one hand, it is something that you would define during the setup, but on the other hand, samples can vary in their quality and integrity, which can totally screw up sensitivity, right? So on the one hand, if you're routinely running this assay in peripheral blood, and if you have very stringent criteria for saying, somebody's white count has to be above X, and we need to use at least X number of nanograms in a reaction, and run X number of replicates or something, then you can protect yourself maybe a little bit from issues of sensitivity that are that are pre-analytical, essentially, where you could have very poor sensitivity if you have no cells to look at, right? But if you have sort of the upfront criteria for your samples well defined, then you can work out some of those sensitivity issues by basically running titrations and figuring out what the analytical sensitivity of your method is. But then it's still always a little bit of an extrapolation, because you usually don't know what somebody's B cell count is in their blood or in their tissue. And so you're still kind of making a guess, and often these methods will report out, clone copies per total PBMC number or something like that, which is kind of ridiculous, because if you have no B cells in there, then you're not really measuring much, right? So
That's true.
and the other challenge is, of course, in a tissue-based sample, it depends a lot on the quality of the sample, both in terms of the amount of sample, the cellularity, the B cell fraction in the sample, but also just stupid stuff like if has it been fixed, or is it just frozen? We love snap frozen samples much more than fixed samples, because fixation will beat up the DNA. And then also the length of time that it sits in fixative matters. So if you're doing a study where you're looking at archived samples and they've been in fixative for many years, that's a bummer, because then your DNA quality will be a lot less good. And then you may have to actually modify your method to amplify shorter sequences. So, our traditional assays use Framework 1 and JH, but for really old, poorly preserved samples, we might need to use Framework 3 and JH. And so those shorter amplicons will, again, if you're looking for a sufficiently long CDR3 sequence, that can work. But if you're looking kind of more broadly at the repertoire and trying to accurately collapse clones, that can sometimes be tricky, especially with B cell sequences, as you know, because they undergo somatic mutation. And so then the V gene identification, when you're looking at very short reads, can be very tricky, because of the mutations in the V gene. And there are a lot of different V genes that might be similar in their distance from sort of the nearest corresponding germline V gene.
So because of these differences in sample quality, it's necessary to run a sensitivity control in each assay. There would be.
Yeah, I think again, a lot of these things don't have so sensitivity controls are tricky. Some assays use like inline controls too as kind of calibrators for the sample quality, but that's also fraught with peril if you have a really poor quality sample, because then your inline control will dominate your PCR and compromise any yield that you get of amplicons from the patient and things like that. So in general, we actually try to avoid using an inline control, but we definitely have a batch control for the run, and we've definitely done titrations, of the assay to sort of measure its analytical performance. And then there's a lot of caveating that goes on in reporting the results, saying things like, this was a limited sample or what have you.
I also assume eventually the sample quality is just so poor that you can't really extract any information from it at all, no matter what you do.
Yeah, one of the most dangerous types of samples are these very paucicellular tissue samples, where people want to know whether there's a clonal expansion. And so there, it's you really have to be careful, because if you only have enough sample for one replicate, you can easily get a PCR jackpot that can look like a false clonal expansion. So we normally try to run at least 2 replicates from everything, even when it's, when you're kind of amplifying almost nothing, because that way you can at least see if you have a reproducible expansion. And what you normally get in a very paucicellular sample is you'll see, these sort of oligoclonal peaks, because that's all there is. But then when you look in the replicates, they don't reproduce. And so that's kind of but again, you caveat this extensively when you're dealing with things like that.
That's good.
Speaking of clones, one of your project is clone tracking and B cell subset analysis in autoimmunity. What is the aim of the project, and how do you envision that patients will benefit from this?
Well, so a subject near and dear to my heart. So, clonal expansions are kind of at the heart of any immune response, right? So, when you have an immune response that if the innate immune system hasn't taken care of things, then the adaptive immune system eventually gets called in to help out and undergoes these kind of cardinal features of responding, and one of them is of course activation, which for a B cell response is typically a T cell dependent germinal center reaction. Clonal expansion, you may have in the case of antibodies, you may have class switching, from IgM to different isotypes such as IgG or IgA, IgE. And also, of course, somatic hypermutation occurs. So DNA point hypermutation of the V gene sequences that encode the variable parts of the antibody. So during this process of clonal expansion, in healthy individuals, these clones will respond very specifically to exogenous antigens that are ideally pathogen derived, and then once the pathogen clears, the immune response contracts, and these cells, for the most part, go away, but you'll still have a population of memory B cells that can be called back into service should you re-encounter the same antigen, and that, of course, protects you from disease. In autoimmunity, of course, things are different, and the immune responses, the clonal expansions, can occur potentially in response to self-antigen, but often we don't know a lot about what self-antigens are actually specifically being recognized, although we do have some pretty nice serology tests for autoimmunity, where there are certain targets that have been figured out. But any patient might have, multiple autoantigens that their immune system recognizes. So it's actually infrequent in patients with systemic autoimmune diseases such as lupus for you to see just a single autoantibody specificity. There can be several. And so the question is, can we somehow monitor autoimmune responses in a way that's perhaps agnostic to autoantigens? Because we can't define what they all are anyway, right? And so one feature of autoimmune B cells, and of any responding B cells, is this aspect of clonal expansion. And with AIRR-C experiments, you can monitor immune repertoires for the presence of expanded clones by looking at their copy number fraction in a sequencing library, right? So, and then you can track these clones using their CDR3 sequence fingerprints, if you will, right, over time. And so the fantasy has always been that, can you identify expanded clones in lupus patients? Can you link them to potentially pathologic B cells, either by immunophenotype or by antigen binding ultimately, but still in the end, can you find these expanded clones, and can you correlate them temporally with disease flares? So, again, half the battle here is finding the right set of clones in somebody, right? And to do that, you really need a series of hooks to sort of enhance the specificity of the clones for the disease, if you will. And right, so if you can do that by some form of antigen enrichment or immunophenotypic enrichment or temporal enrichment, you do that. But at the end of the day, what you hope to find are a series of clones that this patient makes when they get sick, and that you can then use to track their disease status over time, and potentially see if therapy A works better than therapy B based on how their clone sizes change with that therapy. That's one possible use. Another use might be that patient A may have clones that derive from B cells of a particular subset. Like maybe the tolerance checkpoint defect in one patient is early in B cell development, and in another person it's late. Then if you combine immunophenotyping approaches, like cell sorting, with AIRR-C, you can potentially figure that out, right? And then you can map that defect back and say, well, certain therapies might work better for an early stage B cell than a later stage B cell. And if somebody has an early stage tolerance checkpoint defect, then maybe that disease is a little harder to treat because when you keep regenerating early stage cells in an antigen agnostic fashion. Whereas if somebody has a late tolerance checkpoint defect, maybe you have an opportunity to interfere with TB collaboration, or even interfere with a specific type of antigen presentation, and that may interrupt a cycle of collaboration in such a way that you could afford somebody a longer period of disease-free survival. So with the advent of all of these different B-cell targeted therapies that actually are increasingly specific for certain types of subsets, again, fantasy at this point, but still hope is that we could use AIRR-C to optimize somebody's treatment and better phenotype their disease.
It sounds very promising and can be a great companion diagnostic tool. But what are the large obstacles on the way for developing it as a companion diagnostic tool?
Well, maybe the largest obstacle in the case of autoimmunity is that we're not anywhere near close enough to defining these clones. And so that's certainly something that a lot of labs are working on. But I think until we have sort of a set of these clones in hand, if you will, or at least an approach that's robust that we could extrapolate to different patients without having each patient be a massive research project, it's still right now very much in the realm of research. Where I think we're a lot closer to clinical implementation is in the realm of certainly in the realm of B-cell malignancy, but also in the realm of infectious disease. So for example, in infectious disease, there are clones that have been identified that can respond to certain pathogens. An obvious example is with SARS-CoV-2 and actually T-cell repertoire analysis, where there are large efforts underway to define public clones that can identify, that can recognize components of the virus or the vaccine. And potentially this information can allow you to determine whether somebody has an adequate sort of immune status, if you will, or sufficiency of T-cell response and protection to viral infection with SARS-CoV-2. And so there, the idea is you do, a public clone analysis. You look for shared clones that are found in people who have been exposed to either the virus or the vaccine and are not found in people who have not been exposed to the virus or the vaccine. And that's in its most simplest rendition. And this is tricky, right? But then so you basically sequence a whole bunch of T-cell receptors from the blood and you ask, how many public clones are there? And if that number exceeds a certain threshold, you say, okay, we're good. So that's actually being done now. Now, the threshold, of course, the devil's in the details. What is a protective threshold? Well, we don't really know that yet. For T cells, most of the really good data for protection are with antibody responses, where we do have some idea that the level of antibody that correlates with a certain virus neutralization titer, for instance, and that in turn correlates with immune protection. We're starting to have an idea about what that might be for SARS-CoV-2. The T-cell piece of it is still much less clear at this point. But again, for AIRR-C, that type of assay already exists, and there's a company called Adaptive Biotechnologies that has an assay called T-Detect, for COVID, that they have rolled out for this purpose.
So the largest obstacle is to identify the never mind. I think we'll put this out.
The largest obstacle is with these public clones. I think it is very tricky for several reasons. So you can imagine that such an assay would have reasons for being falsely positive or falsely negative, right? So in terms of false positivity, there are certainly certain HLA types where immune responses to the virus are likely to be more robust. and where it also computationally we may have a bigger library of public clones that has been established. So if you sequence somebody very deeply, even a naive individual, you may by dumb luck run into some of these clones. And the question then becomes, at what level, at what proportion do those clones need to be present for that to be a quote unquote positive result? On the negative side, it's even worse, because on the negative side, you could have somebody who has very few T cells, would be a trivial reason why the test could be negative, right? And if they have very few T cells, you may not have sampled enough rearrangements to get a good sense of whether or not they have the public clones. But, and they may well have them, but you haven't sampled them. On the other hand, if they're T cell immunodeficient, maybe it doesn't matter. Maybe they're still at risk anyway, But the other piece to it is that a lot of people, actually most of the immune response that you look at in SARS-CoV-2 to the virus, for example, is private. It's not public, actually. So this test, while relying on, these public clones, doesn't necessarily give you good insight into what an individual's response is, especially if they're a little bit unusual. So if somebody has an unusual HLA type, where their T cell, the public clones that are associated with responding to the virus for that HLA type have not been well represented in the database, then that's another reason you could get kind of a false negative result. So I think it's still early days with this, and it's going to take some tweaking for sure. And in the end, we still don't know, at least for the SARS-CoV-2 testing, how useful it will be, right? Because we're still trying to figure out if you have a nice, T cell response, does that protect you well enough from infection? There are some pretty nice data coming out that suggest that just having a decent T cell response will protect you. I mean, one of the more interesting data sets from my institution is in patients with MS, multiple sclerosis, who receive B-cell depletion therapy. So they have basically very low levels of circulating and even probably pretty low levels of tissue-based B cells because they're under this chronic B-cell immunosuppression. And yet their T-cell responses to SARS-CoV-2 in individuals who've been vaccinated are robust. So yay, and the epidemiologic data in MS would suggest that you're not seeing massive increased rates of infection in that population. But that's with epidemiologic data, you have it's a little bit tricky to interpret that, right? Because people who have a condition like MS also are more likely to be super careful in a pandemic. So, how do we figure that out in the end? It's going to take some time.
Taking figuring out as a keyword for the next question. So if we zoom out a bit and look at things in a much broader perspective, which areas of diseases or other things do you think can benefit from AIRR-C analysis? So in near or also, if we're thinking out loud, in farther future?
Well, I think so. If we kind of put them in order of current to more remote, I would say for current testing, definitely hematologic malignancy. It already exists. It's already being done. And for hematologic malignancy, you have diagnosis of the initial, identification of the clonal expansion, but you also have tracking of the clone with time. And then also as part of identification of the clone, for some of the diseases like chronic lymphocytic leukemia, the measurement of somatic hypermutation in the clone is actually something that's a prognostic feature. So, for example, again, in CLL, patients who have mutated antibody heavy chains in their clone tend to have a better outcome than people who have unmutated antibody genes. So those elements of AIRR-C are already being used in the clinical arena, and I think will continue to be used for both, diagnosis, prognosis, and disease tracking. The second area, which is sort of current but not yet there, is in the analysis of infectious disease responses, such as the immune status of T cells for SARS-CoV-2. And I think that could be expanded to lots of different diseases and different antigen systems and vaccine responses. So that is pretty wide open, I would say, at this point. And one of the really interesting aspects of that is that the thing that will really make that explode is the proliferation of data and data sharing of the type that the AIR community is so fond of, both standardizing but also sharing across different repositories and standardizing the naming of the genes and things of that sort. So as these databases continue to be populated with increasingly dense collections from patients with different immune exposures, and then also correlating those clones to particular antigen reactivity. So, and then having databases like IEDB built up will be hugely useful for this type of endeavor, because then your clinical test for immune status / exposure will not be simply to COVID. It can be to lots of different pathogens and ultimately maybe environmental antigens and other things as well, as these databases improve. And so then you take this one sample, you sequence somebody's peripheral blood mononuclear cells pretty deeply, both their T cells and their B cells, and then you run it through a whole series of algorithms that pick out known public clones for exposures to antigens X, Y, and Z. And then you have kind of an immune profile of exposures and of levels of responding clones or potentially responding clones. So I think that type of analysis is, it's coming. What will feed into it and what is essential for making it work will be the expansion of these databases. So that's on the infectious disease / maybe even environmental antigen exposure piece. And then autoimmunity, the part that, of course, is nearest and dearest to my heart, I think will be probably the toughest nut to crack, just because of the dysregulation in the immune system that you start out with. And again, the databases are going to be very tricky because one of the unique, maybe not unique, maybe that's the wrong word, but one of the problems with autoimmunity is really the dysregulation of the response and the potential for multi-reactivity of the response. So unlike your traditional good response to a pathogen where you have a T cell dependent B cell response and a germinal center reaction and a lot of selection going into that event, and fine tuning of specificity in an autoimmune response, you may have more features of an extrafollicular response, potentially less dependence on T cell help, and also less censoring of autoreactive specificity, with the result that you get not just autoreactivity, but multi-reactivity. And multi-reactivity creates a huge headache for public clone analysis, right? Because how do you link those things then to particular specificities? So there are people who are working on this and trying to create databases of multi-reactive clones as well, and to try to predict features of antibody structures that would lend themselves to multi-reactivity, such as use of certain kinds of charged residues like arginines and lysines, maybe alternating with negatively charged residues and things of that sort. But it's still super early days and it's complicated. So, my big sort of beacon for the future, I think, on the antigen exposure side, as we get better databases, that's going to be so interesting, right? That we're going to be able to do these lab tests where we say, oh, yeah, you've seen these 20 things and you have adequate levels for 18 of them, but maybe you need a booster for these other 2. Yeah.
It sounds like a very nice future for AIRR-C for sure. And with these ideas and thoughts for the future, we are actually at the end of this very first episode of On Air, the podcast of the AIR-Community, with a special focus on clinical use of adaptive immune receptor repertoires. The podcast is supported by the Antibody Society. And if you have any comments or questions, you can drop us a line or tweet using the hashtag OnAir with 2 R's.
Thank you for talking with us, Nina.
Thank you for having me. It was a real pleasure
The next episode will be available in 1 month's time when Ulrik and I will discuss TCRs and cancer with Lindsay Cowell from University of Texas Southwestern. Thank you for listening.